Hello, it is my great honor to introduce our work on discovery of a novel tetrapeptide as glucose homeostasis modulator, with bifunctionalities of targeting DPP-IV and microbiota. GLP-1, secreted by intestinal L cells, plays a pivotal role in glucose metabolism. It offers multiple health benefits, including improved glycemic control, regulation of intestinal motility, suppression of food intake, and body weight reduction. However, GLP-1 is rapidly degraded by DPP-IV in the gut. Currently, the discovery of bioactive peptides with DPP-IV inhibitory activity from natural food protein sources has emerged as a key strategy. However, the lack of genomic and proteomic data for many natural protein resources hinders the efficient identification of novel DPP-IV inhibitory peptides. Additionally, traditional methods suffer from high workload, long cycles, and elevated costs. In this study, we established a high-throughput peptide screening system based on DPP-IV inhibition by integrating molecular docking, machine learning, enzymology, and proteomics with an intestinal organoid model. Using this approach, we identified the bioactive peptide VAMP from hemp seed protein, which exhibits DPP-IV inhibitory activity and modulates gut microbiota, providing new insights into bioactive peptide discovery and glucose metabolism intervention. First, we employed different enzymatic hydrolysis methods to break down hemp seed protein, and the resulting peptide were evaluated for their DPP-IV inhibitory activity. The study found that the fermolysin hydrolyzed peptide mixture exhibited the highest DPP-IV inhibitory activity and effectively increased active GLP-1 concentration. Through a comprehensive assessment of peptide DPP-IV binding affinity, half maximal inhibitory concentration, and surface plasmon resonance response values, the tetrapeptide VAMP was ultimately identified as the most potent DPP-IV inhibitor. X-ray crystallography revealed that VAMP binds to and inhibits DPP-IV by hydrogen bonds and salt bridges, with key amino acid residues. We conducted a 1-week VAMP intervention in high-fat diet-induced obese mice to investigate its role in improving glucose homeostasis by inhibiting intestinal DPP-IV activity. VAMP significantly reduced intestinal DPP-IV activity, increased active GLP-1 levels in both the gut and plasma, enhanced glucose-stimulated insulin secretion, and improved oral glucose tolerance. Similar results were observed in OB mice. We next performed pharmacokinetic analysis and discovered its low oral bioavailability. Even at peak plasma concentration, a substantial amount of VAMP remained in the intestinal contents. Consequently, we conducted gut microbiota sequencing and found that VAMP significantly increased the abundance of the Akkermansia mucinifera. We further demonstrated through fecal microbiota transplantation experiments that the gut microbiota, modulated by VAMP intervention, could also improve glucose metabolism and insulin resistance. Subsequently, by screening bacterial strains, we found that VAMP strongly promoted the growth of Akkermansia. Further transcriptomic sequencing analysis revealed that VAMP treatment significantly enriched the peptidoglycan biosynthesis pathway. Among the upregulated genes, G4O16RS10715, which is a transglycosylase domain containing protein involved in bacterial cell wall synthesis, showed increased expression. In summary, we established a high-throughput peptide screening system targeting DPP-IV inhibition by integrating molecular docking, machine learning, enzymology, proteomics, and intestinal organoid models. Using this approach, we identified the hemp seed-derived tetrapeptide VAMP as an effective DPP-IV inhibitor that is active GLP-1 levels and improves glucose metabolism. Additionally, VAMP promotes Akkermansia growth by upregulating peptidoglycan synthesis-related genes, thereby enhancing gut barrier function.